Next category of biomolecules is enzymes. Enzymes are biological catalysts which are produced by living cells, and enzymes catalyze the biochemical reactions in living organisms. Chemically, enzymes are naturally occurring proteins which are globular proteins. Enzymes are highly specific in nature, and they are required in extremely small quantity and highly efficient. It means they can catalyze large number of chemical reaction. Very small quantity is required. These biocatalysts actually lowers activation energy of chemical reaction. Generally, the name of these enzymes are named after the reaction which they are used. Example, uh, conversion of maltose into glucose units, the biocatalyst is maltase. Conversion of lactose into glucose and galactose, the enzyme is lactase. The conversion of starch into many units of glucose enzyme is amylase. So, name is given like that of these biocatalysts. Now, mechanism of enzyme action, how these biocatalyst or enzyme act. So, actually this theory can be explained by key and log theory, which we have already discussed in surface chemistry. Next category of biomolecules is vitamins. Vitamins are organic compounds which cannot be produced by the body. Hence, they must be supplied in small amounts in our diet to perform a specific biological function for normal health and maintenance of body. Vitamins can be classified as water soluble vitamins and fat soluble vitamins. Water soluble vitamins, as the name explains itself, are soluble in water. These vitamins are stored in lesser amount in cell, hence they must be supplied regularly in our diet. Example, vitamins of group B and vitamin C. Another class of vitamins is fat soluble vitamins. These vitamins are soluble in fat and they are stored in liver and adipose tissues. Examples of fat soluble vitamins are vitamin A, D, vitamin E and vitamin K. Deficiency of these vitamins cause some deficiency disease. For example, deficiency of vitamin D causes rickets, deficiency of vitamin C causes scurvy, etc. The next biomolecules are nucleic acids. So, let us discuss about these nucleic acids in detail. Actually, nucleus of a living cell is responsible for transmission of characters from one generation to the next generation. The particles present in nucleus which are responsible for transmission of characters from one generation to next generation are called chromosomes. And these chromosomes are made up of proteins which combines with biomolecules known as nucleic acids. It means chromosomes are composed of proteins and nucleic acids. So, these nucleic acids are biologically important polymers made up of many units which are present in living cells. Nucleic acids direct the synthesis of proteins and they are responsible for transmission of genetic information. It means nucleic acids gives the direction for the synthesis of specific proteins and transmit characters from one generation to the next generation. There are two types of these nucleic acids. DNA, that is deoxyribonucleic acids, and RNA, that is ribonucleic acids. Nucleic acids are long chain polymers of nucleotides and nucleosides. Now let us discuss about chemical composition of nucleic acid. Hydrolysis of DNA or RNA gives some molecules. These molecules are pentose sugar, phosphoric acid, and nitrogen containing heterocyclic compound, and these compounds are called bases. So when hydrolysis of DNA or RNA is done, then it gives pentose sugar, phosphoric acid, and nitrogenous base. DNA has deoxyribose sugar while RNA has ribose sugar. So RNA has ribose sugar while DNA has deoxyribose sugar. So that is the basic difference in ribose and deoxyribose sugar. Another difference is that DNA has 4 bases: adenine, guanine, cytosine, and thymine, while RNA also has 4 bases. Out of these 4 bases, adenine, guanine, and cytosine are similar to the DNA, while the fourth one is uracil, which is different from DNA. Structure of nucleic acid, as we know that nucleic acids are polymer, and they are polymers of nucleosides and nucleotides. So actually what is nucleosides? Nucleoside is the molecule in which one of the nitrogenous base is bonded with sugar. Nitrogenous base is bonded with sugar, then it is known as nucleoside. In nucleotide, the phosphate group is attached to the nucleoside. So nitrogenous base and sugar is called nucleoside, while nitrogenous base, sugar, and phosphate is called nucleotide. You can see here different structure of this nucleotide, in which pentose sugar and nitrogenous base, and here you can see nitrogenous base, pentose sugar, nitrogenous base, and phosphate. So it is nucleotide and this is nucleoside. The nucleoside and RNA, nucleosides of RNA and DNA differ in 2 respects. Sugar in RNA nucleoside is ribose, while sugar in DNA is deoxyribose. The nucleoside of RNA contains uracil in presence of thymine, which is present in DNA. Formation of dinucleotide means 2 nucleotides are joined together. Nucleotides are joined together by phosphodiester linkage between 5th and 3rd carbon of pentose sugar. So when 5th and 3rd pentose sugar linked through phosphodiester linkage, so you can see this phosphodiester linkage in 2 nucleotides. So there are 2 nucleotides and they are linked together to give dinucleotides through phosphodiester linkage. This is the simplified version of nucleic acid: base, nitrogenous base, sugar, phosphate. So actually this polynucleotide linkage is repeated unit of base, sugar, and phosphate. Double helical structure of DNA. The sequence of nucleotide in the chain of nucleic acid is called the primary structure. Secondary structure of DNA given by James Watson and Francis Crick. Therefore, this model is also known as Watson and Crick model. Two nucleic acid chains are wound about each other and they are held together by hydrogen bond between pairs of bases. The two strands are complementary to each other because hydrogen bonds are formed between specific pair of bases, and these pairs are adenine forms hydrogen bond with thymine, while cytosine forms hydrogen bond with guanine. So here you can see adenine forms hydrogen bond with thymine and cytosine forms hydrogen bond with guanine. So this is double helical structure of DNA. Structure of RNA. RNA is single strand structure. Sometimes they fold back on themselves to form a double helical structure, but it is single stranded structure. RNA molecules are of three types and they perform different functions, and these different types of RNA are messenger RNA, also represented as mRNA, ribosomal RNA or RNA, transfer RNA or tRNA, and they perform different functions. Now, biological functions of nucleic acids. DNA is the chemical basis of heredity, and it is responsible for maintaining the identity of different species of organisms. A DNA molecule is capable of self-duplication or self-replication during cell division, and identical DNA strands are transferred to daughter cell. So, during duplication or replication, same strand is formed, identical DNA strand is formed during cell division, and this duplicate DNA strand is transferred to daughter cell. DNA molecules also synthesize proteins. So here you can say that you can see that replication or duplication of DNA strand. So two strands are formed of DNA. Out of these two strands, one strand is transferred to daughter cell. So this is all about biomolecules which are present in living organisms and living body and perform various important biological activities, life processes in our body, in body of plants as well as in body of animals. So they are very important class of biomolecules. Thank you.